Bismillahirrahmanirrahim. Last time we discussed about structure of the cerebellar cortex. Structure of the cerebellar cortex. Just to sum up, cerebellar cortex is composed of three layers: molecular layer, containing stellate cells and basket cells, and then second layer is a single layer of Purkinje cell. Single layer of Purkinje cell. And the third layer is the granular layer. You see, these are the three layers of cerebellar cortex. Molecular layer, containing the basket cells and stellate cells, and then single layer of Purkinje cell, and then the third layer is the granular layer. In the molecular layer, I told you these two types of neurons are there: stellate cells and basket cells. In addition, there are dendrites and axons coming from deeper layers. The Purkinje cell layer, you see, Purkinje cells are flask shaped. From the top of these cells arise dendrites, which give primary, secondary, and tertiary branches. And these dendrites are in the molecular layer. From the base of the Purkinje cells arise the axon, and when these axons enter the white matter, these become myelinated, and most of these Purkinje cells fibers, these terminate to the deep nucleus cells. Some of these Purkinje cells fibers, these bypass the deep nucleus cell to go to the vestibular nucleus. Third layer is the granular layer, consisting of the granule cells having a scanty cytoplasm, densely staining nucleus. With the dendrites of granular cells, synapse, mossy fibers. You see, mossy fibers, the synapse with the dendrites of the granular cells. The axon of granular cells, these pass into molecular layer, where these have a T-shaped end. And the axons of granular cells synapse with the dendrites of Purkinje cell. So keep in mind, axons of granular cells synapse with the dendrites of Purkinje cells. Now in the granular layer, there are also Golgi cells. Golgi cells, right? Their dendrites, you see, pass into the molecular layer, while their axon synapse with the dendrites of granular cells. So, dendrites of Golgi cells pass into molecular layer, while their axon synapse with the dendrites of the granular cells. This is the structure of the cerebellar cortex. In the cerebellum, there are 30 million functional units. So there are 30 million functional units in the cerebellum. And each functional unit consists of a Purkinje cell and a deep nuclear cell. Right? You see? This is the one functional unit consisting of one Purkinje cell and one deep nuclear cell. I told you last time that in each cerebellar hemisphere in the white matter, there are 4 deep cerebellar nuclei, and these cerebellar nuclei from lateral to medial side, these are dentate, emboliform, globose, and fastigial. So from these deep nuclear cells, nerve fibers come out of the cerebellum, and these are the efferents from the cerebellum. So efferents from the cerebellum are mostly the axon of the deep nuclear cells. Efferents from the first 3 nuclei, dentate, emboliform, globose, these come out of cerebellum through superior cerebellar peduncle. Right? So efferents from the dentate, emboliform, globose, these come out through the superior cerebellar peduncle, while the efferents from the fastigial nucleus, these come out through the inferior cerebellar peduncle. So efferents from the cerebellum, these come out through the superior cerebellar peduncle and also inferior cerebellar peduncle. Right? So I was discussing about the functional unit. I told you each functional unit consists of a Purkinje cell and a deep nuclear cell. There are 30 million such units are there in the cerebellum. 30 million functional units are there in the cerebellum. You see, there are 2 types of afferents to the cerebellum. 2 types of afferent fibers enter into the cerebellum. These include climbing cells, climbing fibers, and second afferent fiber are the mossy fiber. So climbing fiber and the mossy fiber. Climbing fiber and the mossy fiber. These are the afferents which come into the cerebellum through middle and inferior cerebellar peduncle. So the afferent fibers, including the climbing fibers and mossy fibers, these come into the cerebellum through middle and inferior cerebellar peduncle. Climbing fiber are from the inferior olivary nucleus. Climbing fibers are from the inferior olivary nucleus. Climbing fibers give collaterals to the deep nuclear cells. You see, climbing fiber gives collateral to the deep nuclear cells. And keep in mind, these collaterals are excitatory. These are excitatory. And then the climbing fibers pass into the molecular layer, to synapse with the dendrites of Purkinje cell. You see, climbing fibers are from inferior olivary nucleus. These give collaterals to the deep nuclear cells. These collaterals are excitatory. And then climbing fibers go to the molecular layer where the synapse with the dendrites of Purkinje cell. Right? Keep in mind, one climbing fiber synapse with the dendrites of about 10 Purkinje cells. One climbing fiber synapse with the dendrites of 10 Purkinje fiber. So one climbing fiber synapse with the dendrites of 10 Purkinje cells. Dendrites of 10 Purkinje cells. Now we come to the other afferent fibers, the mossy fiber. Mossy fibers are afferents other than climbing fibers, other than from the inferior olivary nucleus. So mossy fibers are all the other afferent fibers other than those from the inferior olivary nucleus. Mossy fibers, which are the other afferents, these also give collaterals to the deep nucleus cell. These also give collaterals to the deep nucleus cells. And these collaterals are excitatory. These collaterals are excitatory. And then the mossy fibers go to the granular layer, where these synapse with the dendrites of granular cells. These synapse with the dendrites of granular cells. So mossy fibers give collaterals to the deep nucleus cells. These collaterals are excitatory, and then the mossy fibers come to the granular layer, where these synapse with the dendrites of granular cells are granule cells. So these are the two afferents to the cerebellum. And keep in mind, climbing fibers give excitatory collaterals to the deep nucleus cells, and then these synapse with the dendrites of the Purkinje cell in the molecular layer. Mossy fibers give excitatory collaterals to the deep nucleus cells, and then these go to the granular layer, the synapse with the dendrites of granule cells or granular cells. Now keep in mind, Purkinje cells are inhibitory to deep nucleus cells. Purkinje cells are inhibitory to deep nucleus cells. And how can we excite the deep nucleus cells? We can excite deep nucleus cells through direct collaterals from the climbing fibers and mossy fibers. We can excite deep nucleus cells through collaterals from the climbing fibers and the mossy fibers. Now keep in mind, one mossy fibers synapse with dendrites of hundreds of granule cells. One mossy fiber synapse with the dendrites of hundreds of granule cells. So this is the neuronal circuit in the cerebellum, and we'll discuss about the functional units. Right? We will also discuss it further later. I told you last time that in the cerebellum, the cerebellar cortex contains the gray matter, and the white matter is in the core. A small quantity of white matter is present in the vermis. A small quantity of white matter is present in the vermis. And large quantity of white matter is present in the cerebellar hemisphere. So in the cerebellar hemisphere, large quantity of white matter is present. White matter of the cerebellum consists of three types of fibers. Intrinsic, afferent and efferent fibers. So in the white matter of the cerebellum, there are three types of nerve fibers. Cerebellum. These pass from the vermis to cerebellar cortex. These pass from the vermis to cerebellar cortex. Or these may pass from one cerebellar hemisphere to the other. These may pass from one cerebellar hemisphere to the other. So this was about the intrinsic fibers in the cerebellum. In the cerebellum, I told you the two types of afferent fibers, the climbing fibers from the inferior olivary nucleus, and the other afferent fibers are mossy fibers. So mossy fibers are the afferent fibers other than from the inferior olivary nucleus. Mossy fibers. So climbing fibers and mossy fibers are the afferent fibers. Efferent fibers. Now efferent fibers start as the axon of Purkinje cells. Start as the axon of Purkinje cells. And then most of these Purkinje fiber, Purkinje cell axon, synapse into deep nuclear cells. Synapse into deep nuclear cells. Deep nuclear cells. These are the nerve fibers of Purkinje cells. The synapse into the deep nuclear cells. Now from deep nuclear cells arise actually the efferents from the cerebellum. So efferent from cerebellum are the axons of the deep nuclear cells. So some of the Purkinje cell fibers bypass the deep nuclear cells. Some of Purkinje cell fibers bypass the deep nuclear cells to go to the vestibular nuclei. And these Purkinje cell fibers which bypass the deep nuclear cells are from the vermis and flocculonodular lobe. So the Purkinje cell fibers which bypass the deep nuclear cells are from the vermis and flocculonodular lobe. Right? This was about the white matter of the cerebellum, containing the intrinsic fibers, afferent fibers, and the efferent fibers. Now about the connections of the cerebellum. Cerebellum is connected to central nervous system or brain through three peduncles. Cerebellum is connected to different parts of the brain or central nervous system. Inferior, middle, and superior cerebellar peduncle. Inferior, middle, and superior cerebellar peduncle. Now I discussed these peduncles and the afferent and efferent fibers in these peduncles. Afferent and efferent fibers in these peduncles. First, inferior cerebellar peduncle, which connects cerebellum with the medulla oblongata. So, inferior cerebellar peduncle connects cerebellum with the medulla oblongata. What are the afferents in the inferior cerebellar peduncle? Afferents are posterior spinocerebellar tract, cuneo-cerebellar fibers, also called posterior external arcuate fibers. Vestibulocerebellar fibers. Vestibulocerebellar fibers. Olivocerebellar fibers from the inferior olivoid nucleus. So olivocerebellar fibers, and then reticulocerebellar fibers. Reticulocerebellar fibers. So 5 types of afferent fibers in the inferior cerebellar peduncle, posterior spinocerebellar tract, cuneo-cerebellar fibers, or also called posterior external arcuate fibers, vestibulocerebellar fibers, olivocerebellar fibers, reticulocerebellar fibers. So these are the afferents, 5 afferents in the inferior cerebellar peduncle. Now efferents. In efferents in the inferior cerebellar peduncle, these include cerebellovestibular, and cerebello-reticular. So cerebellovestibular and cerebello-reticular, these are the efferent fibers in the inferior cerebellar peduncle. Now we come to the middle cerebellar peduncle. Middle cerebellar peduncle, which connects cerebellum with the pons. Which connects cerebellum with the pons. This is the largest of the peduncles. It is the largest of the peduncles. From pontine nuclei, pontocerebellar fibers arise. From pontine nuclei, pontocerebellar fibers arise, which cross over to the opposite side, and then to the middle peduncle, these go to the cerebellum. So pontocerebellar fibers arise from pontine nuclei, cross over to the opposite side, and then enter the middle cerebellar peduncle to go to the cerebellum. Now these pontocerebellar fibers are actually part of cortico-pontocerebellar pathway. So these pontocerebellar fibers are the pathway, are the part of the pathway that is the cortico-pontocerebellar fibers. So this was about the middle cerebellar peduncle. Now we come to the superior cerebellar peduncle. Superior cerebellar peduncle, which connects cerebellum with the midbrain. So what are the afferents? Afferent fibers in the superior cerebellar peduncle. Now afferent fibers are anterior spinocerebellar tract, anterior spinocerebellar tract, rubrospinal tract from red nucleus to cerebellum. So rubrocerebellar tract from red nucleus to cerebellum, rubrocerebellar tract, and then cuneo-cerebellar tract. So 3 afferents: anterior spinocerebellar, rubrocerebellar, and tectocerebellar tract. These are the afferents. Now the efferents from cerebellum, which are in the superior cerebellar peduncle. So these efferents are, that efferent come out of cerebellum. These go to the ventro-anterior and ventro-lateral thalamic nuclei, and then to cerebral cortex. So from cerebellum, efferents go to the ventral anterior and ventral lateral thalamic nuclei, and then to cerebral cortex. Now other efferents come out of cerebellum. These go to the red nucleus. These go to the red nucleus, and then to thalamic nuclei, ventroanterior, ventrolateral thalamic nuclei, and then to cerebral cortex. So some efferents go to the red nucleus, from red nucleus to thalamic nuclei, ventroanterior, ventrolateral, and then to cerebral cortex. Some efferents go to the basal ganglia. Some efferents go to the basal ganglia. So these are the efferents in the superior cerebellar peduncle. Now we have discussed the connections of the cerebellum. And keep in mind that there are reciprocal connections, two-way connections of cerebellum. So there are reciprocal or two-way connections of cerebellum with the cerebral cortex, red nucleus, vestibular nuclei, and reticular formation. So don't forget that cerebellum has reciprocal two-way connections with the cerebral cortex, red nucleus, vestibular nuclei, and reticular formation. So this was about the connections of the cerebellum. Now we discussed about the functional units in cerebellum consisting of the Purkinje cells and deep nucleus cells. Now both the Purkinje cells and deep nucleus cells discharge continuously impulses at a rate of 50 to 100 impulses per second. So both the Purkinje cells and the deep nucleus cells, these discharge continuously impulses at the rate of 50 to 100 impulses per second. I mentioned in this neuronal circuit that deep nucleus cells are excited through direct collaterals from the efferent fibers, connimic fibers, and the mossy fibers. While the deep nucleus cells can be inhibited through Purkinje cells. Through Purkinje cells, deep nucleus cells are to be inhibited. Now in the neuronal, in the cells, in the cerebellar cortex, we discussed about the stellate cells, basket cells, Golgi cells, granular cells, right? Now keep in mind these basket cells, stellate cells, Golgi cells, granular cells are inhibitory. I told you that deep nucleus cells can be excited through direct collaterals from the connimic fibers and mossy fibers. And deep nucleus cells can be inhibited through Purkinje cells. I said Purkinje cells are inhibitory to deep nucleus cells. Right? I also mentioned that basket cells, stellate cells, Golgi cells, and granular cells are inhibitory. We have discussed that granular cells, their axon pass into the molecular layer where these synapse with the dendrites of Purkinje cells. Now when deep nuclear cells are to be excited, this excitation will be through collaterals from the climbing fibers and mossy fibers. But for excitation of the deep nuclear cells, Purkinje cells ought to be inhibited because these are inhibitory. These must be inhibited if you want to excite the deep nuclear cell. And how can we inhibit the Purkinje cell? Through granule cells, through granular cells, we can inhibit the Purkinje cells. And then, if deep nuclear cells are to be inhibited, in that case, Purkinje cells should be excited. How can these be excited? Granule cells which are inhibitory to Purkinje cells ought to be inhibited. And how can these be inhibited? Through stellate cells and basket cells. So granule cells which are inhibitory to Purkinje cells can be inhibited through basket cells and stellate cells. You see the neuronal circuit. Climbing fiber give collaterals to the deep nuclear cells and then go to the molecular layer where the synapse with the dendrites of Purkinje cell. Right? Again, mossy fibers give direct collateral to the deep nuclear cell, and then these go to granular layer, the synapse with the dendrites of granule cells. Right? You can also see that granule cells axon is passed into the molecular layer where these synapse with the dendrites of Purkinje cell. And granule cells are inhibitory to Purkinje cell. Right? And when Purkinje cells are to be excited, granule cell must be inhibited. And these are inhibited by the, say, Golgi cells. These can be inhibited. And they can be inhibited by the basket cells and the stellate cell. These stellate cells you see here, right? So in the neuronal circuit, when deep nuclear cells are to be excited, these can be excited through collaterals from the climbing fibers, mossy fibers. Right? When deep nuclear cells are to be inhibited, these can be inhibited through Purkinje cells. And Purkinje cells are to be excited. For their excitation, granule cells must be inhibited. And then the other cells, stellate cells must be excited. Basket cells must be excited to excite the Purkinje cells. Right? So this is the circuit when Purkinje cells are to be excited. Right? And basket cells excite Purkinje cells. But of course, these inhibit the granule cells. You see the stellate cells, basket cells, right? The granule cells which inhibit the Purkinje cell, and then these cells, stellate cells, basket cells, acting on the Purkinje cell. Right? These can also inhibit but when these can be excited, when these cells are inhibited. Yeah? So this was about the neuronal circuit in the cerebellum. The typical function of the cerebellum, is to control timing of turn on signals to the agonist and simultaneous turn off signals to the antagonist at the start of the movement. I can repeat, the typical function of cerebellum is to control timing of turn on signals to the agonist and simultaneous turn off signals to the antagonist at the start of the movement. Then at the end of the movement, it controls the timing of turn off signals to agonists and simultaneous turn on signal to the antagonist. So at the end of the movement, it controls the timing of turn off signals to the agonists and simultaneous turn on signals to the antagonists. So this is the typical function of the cerebellum. I repeat, cerebellum, it controls the timing of turn on signals to agonists and simultaneous turn off signals to the antagonists at the start of movement. And then at the end of movement, it controls the timing of turn off signal to the agonists and simultaneous turn on signals to the antagonists. Right? This is the typical function of the cerebellum. Okay.